To introduce Dr. Valerian Kagan. He is Professor and Vice Chairman at the Environmental and Occupational Health Department, University of Pittsburgh. He was originally trained way back when in Russia, actually, as a biochemist. Developed an interest in free radical biology, and he's one of the leading experts. And more recently, he moved into the field of oxidative lipidomics, and that's also what he is going to speak about today in regard of cardiolipin. And the title of his talk is Multitasking of Cardiolipins. The floor is yours.
Thank you so much. It's a pleasure to be here. I really love these Barth Syndrome Foundation meetings. And certainly for one reason is that I can talk about my favorite molecule, cardiolipin. Well, we're approaching the date when we'll have to celebrate 75th anniversary of the moment that cardiolipins have been discovered. There are still very many question marks, although a lot has been done. So I will update you today on what we have been doing, and so we'll see how it goes. So now, one of the fields I love to say, like you, that, you know, cardiolipins are all on the inner membrane and something interacts with them and something don't, but we really don't have reliable tools how to see cardiolipins in cells. So this is a picture that was taken just about 3 weeks ago because my colleague, Dr. Dan Yang at the University of Hong Kong, when she listened once to me, she said, wow, I want to do a new probe for cardiolipin, which will be more specific and more sensitive and better than anything that has been done. So that's what they see the products of what she has been doing. So, but still, this is a probe. This is not, we don't see really cardiolipin. We see fluorescence from something that more or less specifically binds to cardiolipin. Well, can we see cardiolipin? I guess that, well, looks like I forgot to load it. Well, okay. All right, so we can see cardiolipin, and you can do it by using imaging mass spectrometry. Well, I wish I put it in, but evidently I forgot about it. So, but overall, you can do direct measurements of cardiolipin kind of distribution and positioning and map it. Unfortunately, the spatial distribution is not as good as here, but you can still see cardiolipin not in one cell, not in single cells, but in many cells, but you can do it. But overall, with this probe, it looks quite good, because if you do it with mitofabs, and mitofabs are proteins that are expressed in mitochondria, you can see that overlap between this probe and mitochondria is pretty good, so it's at least a good thing. Well, let's go a little bit back with cardiolipin. It's a very ancient molecule. We will celebrate 75th anniversary, but actually it's maybe 3 billion in 75 years that we have to celebrate, because cardiolipins are present both in prokaryotic cells and eukaryotic cells. So what's the difference? Well, so in prokaryotes, so the life has been boring after 1 billion years as the planet was kind of first very, very hot, then started cooling down, but it was pretty warm here. So cardiolipins were mostly very saturated and short chains. And Angel can tell you more about this. And then, what a gift from the gods. So oxygen started evolving, and then cardiolipin have changed. So what exactly happened to them? If you look at mass spec and compare the mass spec of E. coli cardiolipins with that of the brain cardiolipins, mouse brain, so you see immediately the difference. Not only the color, but also that this is MZ, so mass to charges. So these are reds, are shorter chain, lower molecular weights, and the blue ones are longer chains, and they're also polyunsaturated. So why is it important? Because here I've heard a lot about oxidative stress, oxidations. We'll talk about this just in a little while. But overall, please note that, of course, cardiolipins are very polyunsaturated, and L4 That's not the limit, of course, of cardiolipin polyunsaturation. So you will see that there are many of them in this part of the spectrum have docosapentaenoic and hexaenoic, so they're very, very polyunsaturated lipids. So now I just want to explain this, but we've heard this wonderful talk about decoding kind of and helping understand this cardiolipin diversity. So with 4 fatty acids, and if they're all four different, so then the number of cardiolipin species, given that we're talking about 32 approximately fatty acids, maybe more. So the total number of species is really goes in millions. Well, that's if they're not oxidized. If they get oxidized, and you can see what happens here, so then you have addition of hydroperoxy, epoxy, oxo, or hydroxy groups to different positions of these polyunsaturated fatty acids. Then you can really develop a huge headache, because the number of individual molecular species goes to tens of millions. So why is it so important? Well, it's important because if you really need to signal something, and if you need a language, the richer your language is, so the more words you may need. Well, is that really true? Well, somehow, evidently, I put a wrong presentation. But anyway, I can tell you that in the languages that are pretty common, if you take English, you go to Webster Dictionary, and there are 500,000 words there. So, and if you look at most European and Asian languages, so 100, 200, 300,000 words. How many do we use? I just spoke with Miriam yesterday that there's a language used in Suriname, Taki-Taki. 340 words. That's enough. We really don't need very many words, at least to explain the basics. So, but for signaling, how many words do we need and how does it go? Well, I just wanted to, in a very brief way, to tell you what we've been doing over the last maybe 10 or so years. And so the results are summarized here as a cartoon. So in normal mitochondria, cardiolipin is confined to the inner mitochondrial membrane. But if membrane gets depolarized, cardiolipin gets freedom to now gets externalized. And then when it's externalized, it can be recognized by autophagial machinery of cells. And particularly this microtubule long chain 3 protein is the kind of receptor of this cardiolipin. It binds these bad depolarized mitochondria and drags them for autophagy, which in this case is called mitophagy. That's a rescue pathway. If it's effective, that's the end of the story, so the cells feel good. But if the damage continues, then we're talking about self-elimination of the entire cell where it happens. So in this case, cardiolipin externalization is not enough. It also has to be oxidized. And so in this way, you get, for the execution of apoptosis, you need transmigration of cardiolipin and its oxidation. And we'll talk about this a little bit later. And then finally, if cells die and intracellular contents gets out, so then that's where kind of extracellular life of cardiolipin can begin. And one of the things that we've noticed that if you take mitochondria with externalized cardiolipin, so they act as immunogenic regulators. And so they're taken up very avidly by professional phagocytes, be that neutrophils or macrophages. So, and independently of this, these cardiolipins interact with plasma membrane TLR4-MD2 molecules, receptors. And essentially what they do is that they cause immunoparalysis. They block production and release of many cytokines. So that's about signaling. So cardiolipins have very many signaling functions. So when we think, oh, mitochondria cannot live without cardiolipins because supercomplexes cannot exist. Is this true? Of course it is true. But it's also true that there are many other functions that are not limited to only these structural roles. All right. So now finally, how else? What else is the modes that how cardiolipins can signal? So cardiolipins during apoptosis, as I mentioned, can be oxidized, and catalyst of this oxidation is cytochrome C. So when this happens, we are talking about oxygenated cardiolipins that can be hydrolyzed. And now it's an interesting moment because the substrates of these phospholipase reactions, they don't require any signaling, additional signaling. They don't require calcium because the signal is this oxygen-containing functionality here. And for this reason, it's calcium-independent. This I stands for calcium-independent phospholipases. They hydrolyze cardiolipins and form monolyser cardiolipins. And then they form, of course, free fatty acids, which are oxygenated. What are these fatty acids? So, of course, you know, understand that monolyser cardiolipins will lead me to talk about Barth syndrome. But now I want to mention that there's yet another group of products, and there are numerous products. And what can they be? Oh, just about everything. You name them. Most of the lipid mediators can be formed from cardiolipins in very unusual locations in mitochondria. All right, and there are some of them, and probably some of them are familiar to some of you, if not to all. Well, as I said, cardiolipins, they always surprise me with new facts and discoveries about them. So, but the old things are also very mysterious and surprising. And it's mostly this, diversity versus uniformity of cardiolipins. Kind of, I call uniform or L4 cardiolipins homoacylated. So why is it so surprising? Well, if you look at these cardiolipins, these are bacterial, of low interest to us today, but these are mammalian cardiolipins. And if you look in the brain, bone marrow, lung, liver, in some of them, like in the brain, in some of the tissues, there is a huge variety of them. But if you look at some others, so there are much larger variety, or there are only few of cardiolipin species in some tissues. So what does it mean? Does it mean anything? Does it mean that some tissues love to signal and some use cardiolipins for mostly structural roles or something of that type? That's interesting to know. But that would be question one. And two, how universal is it? Is it only in kind of Homo sapiens that cardiolipins in the heart are represented mostly by L4? Well, that's kind of, we know that in the mouse model, cardiolipins are pretty diversified too. Well, if you take a brain, so mouse brain has this huge number of different cardiolipins. So then I said, okay, I went to Whole Foods store and bought these 3 species of different fish. So that's one of them. Pretty uniform, they were very similar. So, and you see in the brain very many different cardiolipins, and usually they're even more polyunsaturated. But for Barth syndrome, who cares about the brain? We need heart. Well, here's the heart, so bovine heart. So this is major L4, homoacylated. But overall, there are kind of here in the baseline, and Marcus can probably help me to say how many exactly, 22 we counted, in the bovine heart. So how about fish carp? So it's northern, it's a red snapper, so somebody said that it's northern red snapper. So, but you see in the heart, you have more species of cardiolipin than human brain. So it's not universal, really. So evidently, we are very specialized that for some reason, tetralinoleal cardiolipin has some unique roles. Well, if somebody can tell me what this role is, I will be very thankful. Well, it shows exactly the same thing, that there's only kind of one red in the in bovine cardiolipin. But the very important issue is, okay, how exactly are homoacylated species of cardiolipin, how can they be formed? Because it's a thermodynamically almost impossible event. Why is it so impossible? Well, let me again, so we've heard today about synthesis and reacylations of cardiolipins, and so there are kind of synthesis and then how kind of remodeling is happening through formation of this monolyser cardiolipin. And as I mentioned, it can be formed from cardiolipin by phospholipases of one type and from oxygenated cardiolipin also by phospholipases, but by different phospholipases, calcium independent. So but it leads to the formation of monolyser cardiolipins. And I was thinking about this. This is Pittsburgh, downtown Pittsburgh. That's Golden Triangle, Monongahela, and Allegheny River. And I thought exactly like these two tributaries merge here at the golden point. Monolyser cardiolipin is a golden thing. So we just need to better understand what is it doing. Okay. Well, we know, of course, the story with tafazin and LCAD, MLCAD. So most of the people, the major players are here, and we just realize that this schema is wrong. There may be a number 4 here as well. Well, but that's tafazin is a part of this, what is relevant to. So, but how to do it? How to understand what's going on with these monolyser cardiolipins, and also how homoacylated species are formed. The best thing after I attended this meeting 2 years ago, that I met Miriam here, and she said, Valerian, you will never understand anything if you will not start working with yeast cells, because this is so simpler, and it's like Swiss watch. It happens or it doesn't happen. Okay. So I said, I agree 100%. So how this remodeling takes place, really? So it's a simple reaction. Monolyser cardiolipin then steals one fatty acid from another phospholipid, and this cardiolipin is formed. Wow, there is a price to pay. You end up with lysophospholipid. The number of lysophospholipid does not decrease. You are still ending up with some lyso form, okay? So how to do it in yeast cells? And you have to understand the complexity of this. So this is cardiolipins with heteroacylated. And because if you talk about 32 fatty acids, well, maybe slightly less, but still there is a huge number. And you're talking about hundreds of thousands of molecular species. Now you have to end up with this tabaret that have 4 identical legs. So again, this is now, it's just in single digits, the number of species. How to do it? How can tafazin or anything do it? So in yeast cell CLD1, then we'll form this tri-legged tabarets, and then phospholipid, and then you go this way to remodeling. It's excellent. Well, here's Miriam and I at the meeting that Angela has organized in Florence. And I'm trying to tell Miriam, you know, that you said that yeast cells are the best. Well, here's the problem. If you look at the yeast cardiolipins grown under standard, because we still argue normal or standard conditions in the lab, so then what you see is that, well, there's no polyunsaturated cardiolipins in cells that are grown under these conditions, okay? So L4 is out of the picture, actually, in yeast cells. But all the major attributes and consequences of tafazin deficiency are there, actually. Pretty interesting. You can, of course, feed these cells with something, and this is mass specs in 2 different ways, with either oleic acid, and then you get the major tetraallyl cardiolipin in these cells. You can feed them with linoleic acid. Then you get the L4 story, so to speak. And then you can feed them with many other fatty acids, arachidonic acid. And now you see that this wonderful uniformity and homoacylation with arachidonic acid, it's incomplete. You give the yeast cells docosahexaenoic acid. Well, it doesn't get integrated into cardiolipins at all for some reason. Maybe Roger is correct, at least with regards to yeast cells. So we're working on this, and this is a kind of an ongoing process. But what we've realized on the way, that why yeast cells are so remarkably good, so that in wild-type yeast cells, as we know, there are 2 possible pathways, biosynthesis and remodeling. So now remodeling is due exclusively to CLD1. And of course, Miriam knows how to knock out this CLD1 momentarily. So if you remove CLD1, so then there's only pathway left, which is biosynthesis. So now you can do arithmetics like Marcus has been doing. So you can subtract this from this. And then what will happen is that you will have kind of a remodeling pathway. But the software that we use is kind of a little bit stupid. So what it does, it only plots everything that grows, increases, goes up. And it doesn't do what we put here. So it doesn't show the decrease signal. So you have to cheat on this software and do it, change the signs on this. Okay, now you don't subtract CLD1 knockouts from wild types, but you do it with minus, algebraic sum with minus. And then when you do that, you can make good guesses about products of this remodeling, and then you can also look and see the types of cardiolipins that decrease or maybe potential substrates of this of this hydrolysis. And if you do it this way, so then you can kind of see how from these species of cardiolipin you can get these red ones that are the products of this, and you can certainly indicate their positions in the spectra like this. So it's a very productive process, and we are working on this with Miriam, and maybe next time I will show you much more about this. But the point is that, yeah, we can do many things, but still the question remains. So how can you, using just one tafazine, which is not even specific, how can you make homoacylated species? It's surreal, really. Now, what is lacking is understanding of the structure of tafazine. It has never been crystallized, and it should be. So we started doing, because we are not good in crystallizing proteins. It's just a lot of too much work. I don't like to work too much. So, but what you can do, really, you can do homology models. And so this is glycerol phosphate acyl transferase, kind of a relative. The homology is not remarkably good, but you can dock either phosphatidylethanolamines or cholins as donors on this molecule and see how they position themselves. And you choose lowest binding energies. And then you can dock monolyzer cardiolipin. And by doing so and doing molecular simulations, you can better understand what is going on. But and assess whether any good specificity is possible here at all. But don't expect that it'll be 100,000-fold. That's essentially, I don't think it's possible. So of course, there's also specificity of CLD1. And so assessments that we have done, and we now, as some of the reviewers indicated, we submitted this paper that we need to expand this, and we are doing this now, very good comment. And so that, but you can see that there is specificity. So it's again, there'll be never 100,000-fold. You have to have many factors to get this specificity that homoacylated species require. So, all right. Now, the last thing that I wanted to mention in this series is the experiment that we started with Grant Hedge. This is he loves also this golden point in Pittsburgh where two metabolic streams merge together. So now in this model of Taz knockdown mouse, so we assessed cardiolipins and monolyzer cardiolipins not in the heart, but in the brain. Well, there are many reasons why we need and we love to study brain. But what I wanted to show you here is on this graph. So this is significance, and whatever is above this blue dotted line, that's significant with less than 0.005. And now, and this is it's log 2 of changes of either increase or decrease. And the circles are cardiolipins, and the stars are monolyzer cardiolipins. And this is a scale of absolute kind of amounts. But what I want to show you is this. So that cardiolipins go down. They're to the left from this zero line. So now there are still cardiolipins. It's here. So, but now there are monolyzer cardiolipins, stars. that are they change very dramatically, so manifold changes, but not all to the same degree. So again, if you have only one cardiolipin to choose from, oh yeah, L4 decreases, monolyzer, trilinolol cardiolipin accumulates. But if you have many, you cannot just use one of them to characterize what's going on. And Barth syndrome is not only heart disease. Actually, neutrophils have multiple species of cardiolipins. Certainly, brains have multiple species. And if neurological disorders, as kind of I know from Grant and other people now as I looked at it, so I still kind of have manifestations in Barth syndrome patients. Then to fully characterize what's going on in the brain, you have to study different types, and you really have to understand what you are doing with this. So TAS deficiency affects multiple species of cardiolipin in the brain. And of course, what we were also interested in, and I'm a redox person, I want to see oxidations of something. And we looked very, very thoroughly whether any oxidation happens in brains of these animals, TAS deficient animals. And we found nothing. There is no data because there is nothing to show, actually, on this subject. So again, there are two pathways to form monolyzer cardiolipin, and this is one of them, and is a very important one. And everybody loves to show these schemas that cardiolipins are very close to the sites where ROS are produced. Okay, if ROS are produced, then you have to see products of ROS. Because this is something that I've shown it multiple times in different, but I'm a redox man. I really need to explain to everybody what exactly ROS are doing and how they're doing this, particularly with regards to phospholipids, because I think that there's not complete understanding of this. This is ROS. What is ROS? It's oxygen. When it gets one electron, superoxide is formed. So it's univalent reduction. So the second electron will give you hydrogen peroxide. Superoxide is non-reactive. It doesn't do anything with any molecule except for very few, like nitric oxide, okay? Otherwise, it's not reactive. Hydrogen peroxide is not a radical at all. It's a molecule. It's stable like a brick. You leave it alone. Nothing will happen to it in months. So then the third electron will form this hydroxyl radical. If this is happening, then you're in phantom chemistry. This is the real important reactive oxygen species, which is very reactive and will react with anything. So it will certainly react with lipid, form a lipid radical, then plus oxygen will give you a peroxyl radical. This peroxyl radical, and that's now the key, will react with a neighboring lipid molecule. But the major point is that this reaction is very slow. In saturated lipids, it doesn't happen. In monounsaturated, it doesn't happen, or very slowly. But in polyunsaturated, abstraction of hydrogen from this carbon in the bisallylic hydrogen is very, very effective. So then this cycle can repeat itself many, 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 many times, only in polyunsaturated lipids. Well, that's exactly what happens. And if you do it in a chemical tube, the process develops almost like an explosion. It's very fast, okay? But this is random lipid peroxidation. I emphasize the word random here. So saturated lipids do not get oxidized. Monounsaturated almost do not get oxidized. But linoleic acid containing linolenic, arachidonic, and so on, they are the substrates. And the rate of oxidation will be the greater, the more unsaturated double bonds you have. So essentially, this is what rules chemistry of oxidation in liquid phase, but not enzymology of lipid peroxidation, okay? And because of this, you can, by measuring and analyzing the oxidation products, you can establish whether the process is mainly driven by chemistry or by more specialized and specific mechanisms, okay? And that's exactly what kind of, but this brings us to the field of oxidative lipid signaling and ROS, but please be aware that lipids are among the most susceptible substrate of oxidation reactions. So if you see no lipid oxidation products, you have to start thinking, what is the potential role of ROS in the reactions may be? Well, unfortunately, kind of hunting for oxidation products in lipids is not easy because, you know, so they constitute from 0.001% to 0.1 to 1%, and there's so much more of non-oxidized lipids that you have to be really a good mass spec expert to start doing this. Well, I just wanted to very briefly mention to you that we used 4 in vivo models of different types of stimulation of oxidative stress, and it's exposure of the animals to single-walled carbon nanotubes, and they contain a lot of these iron and green inclusions. And iron is a huge catalyst of phenton chemistry, of production of hydroxyl radical. You expect oxidation. Hyperoxia, just too much oxygen. Radiolysis or exposure to ionizing radiation. So these radicals, different radicals that are formed by radiolysis of water, so they are randomly attacking molecules. You expect chemistry and oxidation of chemical type. And this is traumatic brain injury, and I'll give you just one example. Again, can you show this? You know, I just realized when I put this together, I forgot to put imaging slides, but I realized that as I'll be talking for so long, there was not a single joke, so I want to make one. Can you?
Oh my gosh, hold on.
The model of brain trauma. Can you do the next one?
It's very good.
Very good.
2 months.
3, 2.
So it's a repetitive mild trauma. That's it? I'm out of time? 3 minutes? Okay. All right, thank you so much. So our model is less kind of dramatic. So it's just a rat, but very well calibrated. It's a controlled cortical impact. And so what we see when we did this, that there is no random oxidation. That's the point. So what we see is a very selective oxidation of cardiolipins. So we separate non-oxidized from oxidized in controlled brains after trauma. And what was exciting and interesting that, okay, we got a lot of oxidation products, many species of oxygenated cardiolipins. And we know that in brain trauma there is a lot of apoptosis. And we wanted to understand, okay, which of them are really relevant to apoptosis. And so we did this so that this is apoptosis. Many products, 154, 165 species of cardiolipin underwent oxidation during apoptosis. But which of them are really signals? You cannot get it from brain trauma experiment. We developed what we called optolipidomics. Well, it's known in electrogen orange. You give it to mitochondria, it gets in and oxidizes everything, and you have now oxidation products. Now, if you combine all the data that you collect from brain, from in vivo, in vitro oxidations, where you see apoptosis, you can sieve out relevant versus non-relevant cardiolipin oxidation products. And there are only 3 of them that are relevant to apoptotic execution of apoptotic program. And they are shown here. This is 18-2. So it's one of these L4 linoleic acid residues with monohydroxy group. So these are the signatures of apoptotic oxidation of lipids. Okay? The rest of them may be formed and they signal something. But I don't know what in Taki language, what exactly does it mean. But there are more than 150 of them. So let me just finish with this. So that during apoptosis, cytochrome C, and that's the key. Why only 3? Because it's an enzymatically catalyzed reaction. It's not chemistry. It's chemistry catalyzed by hemoprotein, cytochrome C. Cytochrome C forms a complex with cardiolipin. And in this complex, I just want to show it there. So the reactive species are intermediates of cytochrome C, and they oxidize cardiolipin. And then, of course, it goes to apoptosis. And cytochrome C has very high affinity for cardiolipin-containing membranes, and you can model it and see how it sticks to. So now I really ran out of time. So there are many ways, if you understand the mechanisms and pathways that lead to kind of Barth syndrome, of course you can propose very many different things, and it's very essential to understand whether oxidation does or does not play a role in this, and the jury is still not out, so we have to better understand this. But overall, I think this is a very challenging but also a very exciting moment that we can start doing this. And you can say, well, when are you really going to start talking about Barth syndrome? This is all theories. Well, I agree. But nothing is more practical than a good theory. Thank you.
We run a little bit out of time, but one or two questions. Roger?
Yeah, very quick one. So most important question about your talk is there's a major difference between the living organism with short lifespan versus long. Have you looked at the human or mammals from young to very old? Is there a major switch in the cardiolipin species?
Irrelevant to Barth syndrome, just in humans?
Yeah.
Yeah, we do some measurements in humans. Actually, in humans, you don't have this luxury of having hearts or something, or brains, but you can do measurements in plasma. And in plasma, normally you see very, very, very low levels of cardiolipins. Now, if you have some kind of injuries or disease conditions that may be associated with neuronal damage, for instance, cardiac arrest, then in plasma you can see appearance of either cardiac arrest, cardiac cardiolipins, L4s, or brain cardiolipins. Amazingly, what we see are neural cardiolipins. So we can see, but no oxidation products there.
Okay, last question.
Hi, Dr. Kagan. I know you now. We sent two samples to your labs for oxidized cardiolipin IC using two cell lines, Barth patient cell lines and control cell line. Mm-hmm. We got the result, I remember, very high percentage of oxidized cardiolipin species.
Wow.
I just want to know, this is normal phenomenon or this is abnormal?
If you wish, we can talk. I have to really, because this is a confession that I have to make now, that we didn't make huge progress, because you fed these cells once with linoleic acid, and this linoleic acid was heavily oxidized to begin with. And this oxygenated linoleic acid was readily integrated into lymphoblast cell lines. And we have had so much of oxygenated cardiolipins that you cannot imagine how much. And now we had to redo it, but we didn't really get to this yet. And we discussed this with Michael also. So we will be doing this. It's a very interesting point.
Thank you very much.